Swayam Prabha. Digital India. Educated India.
Okay, what is cooperativity? Lot of slides are there, it's written form, I will explain very briefly first here. Okay, the moment one of the oxygen binds like this, okay, how it binds? It binds like this, iron, porphyrin site, iron, porphyrin site, this is imidazole, that's the deoxyhemoglobin, no oxygen is there. This is the other histidine that I am talking over here, which is far, okay, this is the reduced form, oxygen binds with it, and what happens, this is the oxy form, oxyhemoglobin, and there is a hydrogen bonding. Okay. Now the moment it binds, this is definitely going to come one form or the other in the exam. What happens? This iron in the deoxy form is in reduced form. Okay, both the structure we are discussing only iron 2 plus, no iron 3 plus, iron 2 plus. Iron 2 plus, see this is not a very good ligand, it is too far, it is not going to bind. You have a porphyrin ring and a imidazole. This is iron 2 plus high spin. It's a high spin configuration, and this iron is not really in the plane. So if the porphyrin plane is here, iron is sitting half in, half out. It's not into that cavity perfectly. So as you can see the iron on top. It's of course little bit in the cavity, but it is on the little top. Okay, it's not really inside the porphyrin cavity. What is the porphyrin cavity we were showing here? This is the cavity, right? Now this is iron 2 plus in high spin form. The moment oxygen binds, okay, the moment oxygen binds over here, it goes to low spin form. So it's now a octahedral complex. Perfect or not perfect, it's a very good octahedral complex, right? And you know that iron 2 plus high spin means what? T2G4 Eg2. Upon the oxygen binding, it goes to T2G6 Eg0. T2G4 Eg2, this is T2G6, high spin to low spin configuration change. The moment high spin to low spin configuration change is there, so there is T2G Eg, all of a sudden there is no electron in Eg, and therefore what happens is the repulsion of the ligand becomes less and the size of the iron decreases. So in this case iron 2 high spin is larger in size, low spin is little bit shorter in size. Little bit shorter, that's good enough. That means it will go into the cavity in the porphyrin, and thereby there is that pull, that is a structural change. So this imidazole, you can see it was bind, or it is bind with the iron center, and it's like big, iron center is big. The moment it gets shortened, it just, so it's over here, iron is here, you make it little shorter and then it pull in. That change, it is over here, it goes down. That change will trigger the cooperativity. What will happen? The moment it gets pulled in the cavity, that change in configuration or conformation will be relayed all over. That will send the message. The moment one oxygen binds in here, it pulls in or it passes the information and this guy knows that oxygen is available, and it almost again this guy undergoes high spin to low spin configuration change, oxygen comes and sit down. It happens to here, it pulls in, again it relayed. So the activation barrier or how much energy you need to put for the fast oxygen is always going to be high. The moment you are able to bind the fast oxygen, second, third, fourth is going to be less and less. It's just a relay. I mean, you are having a 1600 meter race, let's say. It's just gets relayed very fast. If there is a message that oxygen is there, the high spin to low spin change is happening. Oxygen binds, high spin iron 2 plus goes to low spin iron 2 plus, it gets reduced in size, it gets pulled in the pocket of porphyrin. That little change, it's a very subtle change, it gets communicated with the next porphyrin center. That next porphyrin center binds oxygen, it gets communicated next one, it gets communicated next one. So that is cooperativity. They are cooperating, they are working in tandem. They are very good communicator. They are communicating with each other. Okay, now that's cooperativity. It's very nicely also written in the slide form. I will I may not discuss way too much afterwards. Okay, now so how the binding happening? This is oxygen, these are, let's say, these are oxygen 2 electrons. It is ligand to metal bonding is happening. Okay, it gives to the dz2 orbital, the axial position, right? It gives to dz2, and from dzx orbital, it donates back to the oxygen pi star orbital. Right? So it's a back bonding. It's forward bonding and then back bonding. That means that the binding is going to be good. It's of course, it's not too tight, but it's good. Iron center will bind with the oxygen decent amount. That is occupied, means field 1 will be donating, and then there will be back donation. Okay? We'll not get into too much details of that. Now this is what exactly I tried to say. I'll just briefly go through the slides. It will be provided to you in the Moodle, so I'll be going directly. Now iron 2 plus, this is high spin paramagnetic. Of course it is high spin paramagnetic, right? Because it is T2G4 Eg2, high spin, right? Iron 2 plus paramagnetic, T2G4 Eg2. The moment oxygen binds, it becomes low spin diamagnetic, T2G6 Eg0, no unpaired electron. Yeah. No, it's not really octahedral, but it's but still there is a loose coordination. As I was showing, this histidine is there. The moment it is there, it is, of course, the 6th coordination side is almost vacant. It's not 100% vacant, almost vacant. Okay. Now, of course, you can see that this information, iron 2 plus 2, this iron 2 plus, I mean, not really deoxyhemoglobin to oxyhemoglobin changes, this will also have some effect in the spectroscopic behavior. Okay. What we are not discussing here is right after oxygen binding, iron oxidation state also changes. Iron 2 goes to iron 3 plus, oxygen becomes superoxo, reduced by 1 electron. That we are not discussing. You don't have to write in the exam. Do not write in the exam. Oxygen gets reduced by 1 electron, that is called superoxo. What we have discussed here is paroxo. And iron 2 plus gets oxidized to iron 3 plus. That is a reversible process. Means it can go forward and then come backward as well. Anyway, now due to all these changes, we will get a spectroscopic difference. The color, or if you measure before oxygen binding and after oxygen binding, the spectra of the compound will be different. Based on that, we can measure how much oxygen is bound. Let's say some people suffer from this less oxygen delivery to certain part of body. That is bad, right? So maybe enough porphyrin center are not there. Okay. So how much, let's say at a certain body part, how much oxygen is there, that you can calculate. It's a very simple test. You take the blood, or you take some site-specific reaction or site-specific study, a specific area you just see, or in general you can figure out how much is there. So how much oxygen content is there from this spectroscopic change, you can find out. That is what pulse oximeter is. Okay, you can just read and understand, nothing in there. Now, what happens when oxygen binds to hemoglobin? I think I have discussed already. So high spin goes to low spin. Okay, all the information is covered. So this phenomenon is called cooperativity. For your exam purpose, these are the slides, the exactly same thing what I have said, without looking at this slide, those informations are there. You read it. Okay. Now, see, there are some slides. I will take 5 to 10 minutes to discuss. This is for your general understanding. Well, the problem is, in 1 minute, let me discuss the problem. So all other section, whoever are the instructors, they have discussed it. Okay. So I have to discuss it. Okay. Simple. But I will give this slide in this form, so you decide how much you want to read, how much you don't want to read. I have given, it's for your information. I'm very clearly saying, I think we are not going to, most likely, 90% guarantee. Maybe some information, understanding may be tested, but not in deeper level. Okay? Let me get into that. You will see it's not that boring. Okay? All right. All, let me go to the next slide. It's interesting, right? Right. All right. We do know that our blood is red. Then there are some animals, some other species who has blue blood. These are not demons, not in movies, the blue-blooded. There are cold-blooded people, okay, but these are blue-blooded animals, okay. So the reason why, this is again not for exam. Give me 5 minutes, I will finish it. It's important that you understand, okay. Never you are going to look back perhaps ever again these things, okay. It's important you just do understand a little bit. I'll discuss some little bit interesting thing, okay, including daru, okay, how it get digested. Okay, so in our blood we have this porphyrin center, right, iron center. These other cases, in lot of other animals or some of these these marine invertebrates, they do not have the blood or so-called blood as we see in red, because they have, of course they have to take oxygen. All we are saying is these are the oxygen carrier protein. The way they carry the oxygen is little bit different. Instead of iron center, there you have copper centers. Those or these porphyrin iron center, instead of those you have copper centers. That gives it blue color. So there are some of these hemocyanin, or some of the animals are there, some of the species are there which have blue blood, right. At the end of this class we will be discussing tutorial part. These are the one where we have, again slide will be given. I think I may not be allowed too much, but it's okay. So there you will be saying the blue blood, okay. There you will be seeing the violet, okay, violet pink color. due to the fact that the ligand is different or the metal is different or both is different. Okay? Therefore, although oxygen binds, mode of binding is different. Okay? Now, these are the slide, amino acid, peptides and protein. You know it. Amino acids is something like this, acid and the amine part. Amino acid put together is protein backbone, right? And these are different amino acid we are talking, aspartic acid, glutamic acid, lysine, serine, cysteine, lot of, I'm not asking you to memorize, but if I think 3-4 amino acid you must know. This is your responsibility. If someone is asking question, I cannot help. Okay, so few amino acid you must know, and you know that's how the peptide backbone is formed. And then those peptide backbone can have the hydrogen bonding or interlinked or cross-linked, and thereby they can form the 3D structure. Okay, that is, I think you have studied in biology or somewhere, or you just essentially understand how it forms. Fine. Next is enzyme substrate complex. I will take you here. Enzyme substrate complex is what? You have an organic compound, organic you have taken, you see a lot of carbon hydrogen bonds are there. And if that organic compound is getting functionalized, let's say hydroxylated, lot of possibilities are there, right? Only a particular carbon hydrogen bond gets functionalized. How does that happen? That happens because, let's say you have a porphyrin center or a iron center, in front of that whole organic molecule is there. It's not like that. Only a particular carbon hydrogen bond gets exposed or comes very close to the active site, that metal active site, metalloenzyme. And therefore, even if you have all those carbon hydrogen bond or carbon hydrogen bond, covalent bond present, all those hydroxylation products are possible, only selectively one carbon hydrogen bond gets functionalized, because that's how it gets exposed. Not these are getting functionalized, only let's say this is getting functionalized. Hydroxylated, you see only one particular CH is getting hydroxylated. Now that's called substrate binding pocket. Substrate comes in, there is metal binding pocket, there is substrate binding pocket. Substrate comes in and sits in there, and the metal is there and the chemistry happens. Okay. Now, so enzyme and substrate interaction, those ES complex we are saying, this is where the substrate is bound by relatively weak forces. Substrate is not going to bound, that organic substrate, let's say, is not going to be bound very strongly. They forms weak complexes. Okay. Another important thing is to understand enzyme is huge. Not everywhere it is binding substrate. There is only a specific center where it binds, right? Now, rest of the things may not be involved in catalysis. This is the reaction we are trying to tell is catalysis, right? It's a reaction happening. Rest of the protein backbone or peptide backbone, we are not seeing much of a role in the substrate binding. Only a particular site or particular place, substrate comes in, sits in there, and the enzyme or let's say metalloenzyme does the chemistry or the catalysis part. So something about the enzyme substrate complex is given in the slide. It's a very easy to understand. Once again, I do not expect you to memorize all these, these things. These are not of much of use for the course purpose, but you try to flip through, give yourself 10, 15, 20 minutes. Some idea wise, if some question is coming, you should not be complaining. We are not going to ask you to draw peptide bond, okay, right? Anyway, now let's move on. This is one of the things I was trying to tell you synthetically. See, if this is the umbrella like structure, you see the umbrella is forming over here, okay? Now oxygen is prevented in the synthetic system. Oxygen is, that's how prevented from reacting at the other center. That's just a view. You see that how people make the porphyrin functionalize, from porphyrin backbone, how people put the umbrella and put the oxygen in a protected manner. It's protecting. Okay. Hey, whoever is not interested, please be my guest, leave it. Okay? Or don't be my guest. Now, carbonic anhydride. There are certain enzyme, okay? This is called, one of the enzyme called carbonic anhydrase that has zinc site at the middle. Now, it is coordinated with histidine, histidine, histidine and water. What it does, it converts carbon dioxide into carbonate. Okay? How it does, again you do not have to memorize, you just see one time. It's a very easy organic chemistry I am talking about. You have zinc hydroxo formation, zinc water was there, okay, and 3 histidine was there. These are the ligand. For zinc 2 plus, zinc is redox inactive, zinc 2 plus stays zinc 2 plus, water gets deprotonated. Water itself, deprotonation will not be easy, but the moment it binds with this Lewis acid, zinc 2 plus acts as a Lewis acid. The acidity, it becomes highly acidic, this OH bond. Deprotonation becomes easier. Water itself, under the physiological condition, it will not, let's say, undergo deprotonation. Now, the moment water binds with the zinc 2 plus is a Lewis acid, and thereby you will have an opportunity to deprotonate. The moment the hydroxy forms, carbon dioxide can be attacked by this hydroxy, and then you can form the carbonate. So, see, sodium bicarbonate, it's something like that. So, carbon dioxide forming carbon bicarbonate or carbonate. Okay? So, that's the carbonic anhydrase activity. Carbon dioxide reacts with water, it's a simple reaction, but it's not that very easy at all. I mean, you take carbon dioxide gas, put water, how can you react? But simple zinc site can do the wonder. It's catalysis. Very, very good. I mean, these are all happening in our body, man. Come on, it's very important. Okay, now then the peptide back bond cleavage. Of course, you know how to cleave a amide peptide bond. It's an amide linkage, right? Hydrolysis you need to do. But this hydrolysis in organic chemistry, what you learn, it may not be efficient, may not be fast. Okay, it's slow enough. But the moment you have this zinc site, this cleavage becomes easy. Okay, it cleaves actually in a similar manner. So this hydroxo, this zinc hydroxo that forms over there, this zinc hydroxo attack over there. Of course, let's say base catalyzed hydrolysis of amide is possible. It's a amide bond is there. You put a base, a nucleophile, attack at that center, and it gives you an acid part from here and amine from here. Right? It's a hydrolysis, ester hydrolysis you have studied. It's a amide hydrolysis, but that amide hydrolysis is very slow under physiological condition. But the moment zinc site is there, zinc hydroxo forms. Due to zinc being the Lewis acid, deprotonation becomes easier. Deprotonation of water becomes easier. That zinc hydroxy can do this wonder. Okay, if you drink alcohol, your liver will be damaged. Why? It's very simple reason, right? Your liver is going to be damaged. That's mainly all. First, to remind you, ethanol is the one which is used as alcohol to drink, not the methanol. The methanol you drink, again I will see you, okay. All right, so why ethanol, and then how our body digests it or breaks it? It's not good for our body. It's a foreign object. We get high, but that's why we drink. Okay, but well, not necessarily. I do not promote, just like movies. Any actor does not promote any of these. I do not promote anything. I am not promoting anything, and that's a very serious note. It's an academic institution. Okay, now, so how? Although definitely people drink alcohol, that's a fact. How body digests it? What happens? Alcohol, ethanol, CH3, CH2OH. Ethanol is converted to acetaldehyde, CH3CHO, alcohol oxidation to aldehyde happens, and then aldehyde gets chopped off to carbon dioxide and water and so on. That's another enzyme. The first step, ethanol to aldehyde formation, that happens in here, the enzyme called LAD, liver alcohol dehydrogenase. It's nothing but, once again, it's a zinc enzyme. Okay? Alcohol, if, that's why liver gets damaged. If the enzyme is not working, which is at the liver, that enzyme is not working, of course, our body will not have the ability to dispel dispel the alcohol from our body, right? If liver alcohol dehydrogenase is not working, that enzyme is not working, alcohol becomes toxic for us. I mean, of course it's toxic, but it becomes even more toxic, right? It could be deadly. I mean, if someone has, it is deadly. Yeah, you do see the news. Someone has liver which is damaged and keep on consuming alcohol, of course, right? So this is how it happened. It's a little bit different mechanism. I will not, you don't have to remember again. What happens over here is, this is the pyridine ring you are seeing. It's called nicotinamide, well, something like similar moiety is there in your cigarette also, nicotine. Okay, all fine, let's stop there. So this hydride, so zinc center binds with the alcohol, R is CH3, methyl CH2OH now gets deprotonated, and this hydride gets transferred to this pyridine ring. It's a, this is the center which is helping, and the enzyme is zinc enzyme, okay? Zinc center with 3 of them, zinc center with all those coordinates, and the alcohol bind in here, and the way alcohol is converted to aldehyde is shown is there. It is a hydride transfer mechanism, simple organic reaction. But without this enzyme, you can understand this process will not happen, and it doesn't happen. That is also true for a lot of these East Asian people, like Chinese people, Korean people. You see, the moment they drink alcohol, their whole body goes red. Not all of them, some of them. Fortunately for us, most of us are, a lot of us are, not most of, a lot of us are brown, so we do not show up that much. All Chinese, lot of Chinese people, lot of Korean people, South Korean people, as far as at least I have seen, actually they do not have this enzyme sufficient amount. Of course, it goes back to your gene. It goes back to your evolution or your generation, how it has come. See, that's how, if the enzyme is not there in enough quantity, what will happen is they are not able to digest or take much amount of alcohol. Little bit is good enough for them. Their body gets swollen or red color. It's just like almost like an allergic symptom it showed up. Of course, some of us can have the exactly same symptom if your body or if your liver is not having this enzyme. Anyway, enough for that. So this is some information about the cisplatin, how much money you can make. You can be a trillionaire, a billionaire, dollars, not rupees, okay? You can be, of course, rupees is good enough. You can be, if you have discovered a good drug, if you have discovered a good catalytic reaction. Simple thing I didn't discuss, let's say water to hydrogen production, that's another enzyme. Since the class is only one, we didn't discuss that. There is an enzyme which can convert water to hydrogen, right? Water splitting. Now, if you happen to have a good catalyst for that, this is fuel, right? Fuel, lots of fuel. Let's say if you have an enzyme, there is enzyme, if you can convert, let's say methane to methanol, that's $1 trillion project. If you have a good catalyst, enzyme does this thing routinely. Enzyme is very good at doing this. Methane to methanol. I'm giving you target. You pick up, be trillionaire, give me some money back. Okay. These are ideas. Okay. Methane to methanol. You produce hydrogen. Okay. This lot of simple reaction which nature does. It could be in our body. It could be somewhere else. If you can develop a good system, catalytic system for them, I mean, nothing can stop you from being rich. Okay? It has to be efficient. Okay? It has to be industrially viable. Industry has to buy your technique. Chemistry can be rewarding too. Okay? All right, tutorial. Water storage and transport protein, we have discussed. Storage protein stores things, transport proteins transports things. The answer is given over here. Answer is given here. If you give me an opportunity, I will wait for you. Why cyanide is toxic? We have discussed again. The binding site over here for oxygen will be occupied by cyanide, and thereby it is toxic, and you cannot, that's an irreversible process. You cannot change it. Okay. So what is the role of globular protein in oxygen transport? It's nothing. It prevents the dimerization. It prevents oxygen, iron center to get further oxidized, and other side reaction it prevents. Okay. Lot of side reaction can happen, that can be prevented by this globular protein. What is cooperativity? Everybody knows by now, right? If you didn't understand, go through this stepwise. Okay? It is fine. Now, why are all the oxygen carriers that contain iron and porphyrins found inside the cell? Why inside the cell? Outside the cell is very oxidative atmosphere. Iron can be oxidized irreversibly to iron 3+. Then you cannot have them used for oxygen carrier, right? Why is the size of high spin iron 2+ is larger than low spin iron 2? We say that high spin to low spin, T2G4 Eg2 to low spin you are going to T2G6. Eg is out. Eg out means lot of repulsion is out, and the cell is becoming smaller, or sorry, not cell, iron center is becoming smaller. High spin to low spin, size varies, size gets smaller. Okay, it is written over there. What prevents synthetic iron porphyrins from functioning as a oxygen carrier? We have discussed this umbrella thing we are talking. Don't write umbrella, okay, and this is just for explanation. You can write. If I am checking, you can get marks. If I am not, you don't get marks. So, okay, it prevents dimerization. Dimerization we have talked. Iron 2+ and 2 iron center can give one electron to the oxygen. Oxygen will be reduced to peroxide, and this dimer formation. See, some of these terms you must write. If this question is asked, the first thing will be it prevents dimerization. Even if you explain a lot of things, you don't say that it prevents dimerization, you are not going to get the mark, hopefully. Okay. Why CO is toxic? We have discussed. Yes, you know that. While the cisplatin is potent anti-cancer agent, its trans isomer is not. Yes, that is cisplatin, that is transplatin, and how it binds we were discussing, right? So see, the 2 guanine, for example, are binding. So actually, sorry, here actually chloride is going out. Okay, all right. Okay, so this is. Are you convinced? I don't think so. Okay, are you convinced with the statement that the coordination complexes are capable of acting as drugs? I think you better be convinced. Okay, there are medicine, I'm sure some of you are taking, or some of us are taking, all of us are taking at some time of our life, which has metal in it. Okay, I think, I forgot. I do have jokes, but I cannot tell you now. But I'll leave you with this. Okay, this is the last slide. A message from, oh, right.